In this video, I discuss gene list submission. The first place to go for information about gene lists is to the Help page. Under the Help tab, we find the gene list submission page. In here, we can find documentation that provides all the details required to submit a gene list successfully to CGAP. One will also find template files available for download here that then can be used for your submissions. Taking a look at a couple of gene lists I've created here, we see that I have two files, one an Excel file and one a text file, and either of these are acceptable for gene list submissions. These are also the only file types acceptable for gene list submission. Gene lists are relatively simple documents, mostly consisting of a title and a list of genes. If we focus on an Excel file, we see that we use headers in the first row, title, genes, and cases. Cases here is actually optional and not required for a gene list. For title, we only expect one value. For genes, you can put in as many values as you'd like. And cases can be empty, non-existent, or have a list of the cases for which you'd like the gene list to be associated. Similarly, for a text file, we expect a title given by the word title followed by a colon, cases followed by the word case or cases followed by a colon, and then a comma separated value of case accessions, and then a list of genes with one gene per line. For either file type, the genes given can be gene symbols or aliases, ensemble IDs, NCBI gene IDs, or other IDs. See the documentation for all acceptable identifiers. Returning to a case, I can find the case accession available either right under the title, and here I can copy it to my clipboard, or I can find it on the details page under accession. And again, I can just copy it to my clipboard. I'll use this accession for this gene list. And so I will copy and paste it and save here. Putting a case accession on a gene list enables the gene list to only appear for those requested cases. In this case, in this situation, just this single case. If I take a look at the two cases of interest here, I can see that I have some gene lists already available. And similarly, I have some gene lists available in this case, though I do not have the gene lists I've created in the other window. I'll now go ahead and submit these gene lists. And to do so, I will use the web UI interface. To find the web UI, I go to the home page of my deployment, hit the Submit New button, and select gene lists. Here we find the institution available as well as the projects available. If you only have one project, it may already be chosen for you. And I will proceed to upload my gene list by choosing my file and hitting upload. In the background, what's happening is we're processing the genes that you provided into the genes in our database. It's important to note that not all genes from Ensemble, for example, are in our database, and currently our database consists primarily of protein coding genes. I see that this gene list passed, it validated successfully, and I did have a gene list created, as I can see here. I had 36 genes and all were found. If I look at the gene list, I can see all 36 genes with the names given, with the names here being the names in our database, not the names that were provided by the submission. Now, if I go back to a case, the variants will start being updated with that gene list if they have genes on that gene list. However, this process does take some time. So for especially large gene lists, it's best to wait a good period of time before starting to use the gene list as a filter, as not all variants may be updated yet. This is an important point to keep in mind, and it's best to submit gene lists ahead of time when they're actually required for filtering. Now I'm going to refresh this case, start a fresh filter block, and see if I have any genes on this gene list that are starting to appear. And indeed I do. I have my congenital glycosylation disorders, and here are some variants with genes that were on my gene list. Now I'm going to submit my text file gene list, which again has only one case associated with it. I go to my home page. I click the Submit New Gene List button. I have my project set up. I find the file. And I upload this gene list. Again, it's going to take a little bit of time to sort through all those genes and match them to things in our database. But I expect the submission to pass successfully again, and indeed it did. And now I see I do have a case associated with the gene list. If I take a look at that gene list, I can see, again, the genes available. And if I go to my case, I can refresh my case and see those variants starting to be associated. I'll start with a fresh filter block and look for gene lists. And there is the glycogen storage disease gene list I just uploaded. Now, if I go to a second case on my project, this one was not associated with the gene list, but was associated with the Excel gene list I submitted. If I refresh it, start with a fresh filter block, and take a look at the gene list, you can see the congenital glycosylation disorders gene list, which was that Excel file submitted, is associated with variants on this case. However, the text file which was submitted is not associated with this case.